I thought I'd begin by thanking Davor and Mike for this really great series of webinars, which I think has done great things for the ISHR and also for the community in these difficult COVID times. We've heard some great scientific presentations, but I want to change gear today and talk very much about the seamier side of science. And I want to talk about the idea that problems in science, scientific fraud, and indeed may be helped, may be encouraged by the system. Sorry, I'm just going to like problems are. So I want to ask the question, does the system reward scientific fraudsters? And in general, does the system discourage reproducibility? So this is how science is supposed to work. These are two of our PhD students, and they're now sitting on each other's shoulders. And of course, this is just a diagram of this famous quotation attributed to Newton: "If I have seen further, it is by standing on the shoulders of giants." So the idea is that somebody does an experiment, reaches a conclusion, and somebody else then develops that, and so science progresses. But there's very much a realization today that science doesn't always work in such a smooth way. And one of the people who've written to this at great length, who I recommend to you, is John Ioannidis, who's written articles with titles of which are "Why Most Published Research Findings Are False." And the ideas behind this come from various sources, one of which is the pharmaceutical company Bayer. And of course, being a drug company, the drugs they produce depend ultimately on basic science done often in university labs. And Bayer reported that they could only reproduce 25% of preclinical studies. Another company, Amgen, said they could only reproduce 11% of what they call landmark studies. And this, of course, is very worrying. Others have suggested that part of the problem comes from poor quality preclinical animal studies, leading to false leads. Very worrying. Now, of course, if you step back for a moment, it would be naive to expect that science should be 100% reproducible. Science is, of course, a human activity. And indeed, many scientific advances have come from lack of reproducibility. So, for example, in my own field, the field which Mike works in as well, the fact that we know that calcium is required for the heart to contract comes from Sidney Ringer and the initial mistakes of his first experiments. But nevertheless, there's great concern that science is not reproducible enough. This quotation here: "A growing chorus of concern from scientists and lay people contends that the complex system for ensuring the reproducibility of biomedical research is failing and is in need of restructuring." Worrying enough words, even more so when you realize this is a quotation from Francis Collins, who was director of the NIH, has probably the biggest budget for research in the whole world. So if he's worried, we should all be worried. So an obvious question is, what causes the lack of reproducibility? And in the next 25 minutes or so, I want to focus on two broad areas. One, the main topic of the talk, is scientific fraud, and the other is poor experimental design, execution, and analysis. And although I give you these now as two separate areas, perhaps at the end of the talk we may come round to the idea they're not as different as we think. So what about fraud? Well, there are lots of examples of fraud that you probably know about. One of the earlier examples of scientific fraud was Piltdown Man. Back in 1912, people were interested in studying evolution in the link between apes and man. And Piltdown, which is in Sussex in the south of England, it was reported they found this skull which looked like a mixture of an ape skull and a human skull. And it was not until 1953 that this was finally debunked. It was a crude forgery where someone had taken an orangutan's jaw, shaved off the teeth so it looked less orangutan-like, and connected it to a human, the rest of the skull, of pure fraud. Another famous example of that of the field of skin transplantation. As you'll know, if you take the skin from one animal and try and transplant it onto a non-isogenic animal, it'll be rejected. It was reported that you could take skin from a black-skinned animal, transplant it onto a white-skinned animal, and the transplant would not be rejected. But it was subsequently revealed that what the scientists had done was to just take a white-skinned animal and paint with a black pen on it to make it look like skin had been transplanted. Pure fraud. There are many examples which you'll know about of what I call dodgy gels. Here's an example. These are points at 4 different time points. Look at the C-kit one here. If you look at this gel, you can see this blot here. You can see a little mark here on the left. You can see one down below here. If you look at this one here, you see exactly the same protrusion there and there. In other words, what had been done here was to just copy and paste one from here to here. It doesn't just happen in gels, it also happens in electrophysiology. Doesn't really matter what this is. Control and one drug, control with 2 drugs. These are separate single channel sweeps. What I want you to notice is that these 3 here outlined in red are identical to each other. And if you look at this one here, normally done on a completely different occasion, this here is identical to this one here. Again, fraud. Fraud continues today. This is a couple of less than 2 years ago. One of the major universities in the United States has had to ask for more than 30 papers published from one group there in the cardiac stem cell field to be retracted. And another very distinguished university has had major financial problems as a result of grant funding given for fraudulent research. So an obvious question is, how common is fraud? And it's an easy question to ask, but a much harder question to answer. Very much a minimum estimate, 0.02% of papers are retracted because of fraud. But of course, this ignores the fact that sometimes fraud isn't found out. And even if it is found out, papers aren't retracted. So as I say, it's a minimum estimate. The Journal of Cell Biology several years ago reported that 1% of papers submitted to them had improperly manipulated images. Again, probably very much a minimum estimate. So what has been done is to send people confidential questionnaires, and the meta-analysis was done by Finelli. I'm amazed this works. What's done is you send a questionnaire to researchers and ask them whether they themselves have ever committed research misconduct or if they know of anybody who does. And interestingly enough, 2% of scientists admitted that they themselves had carried out major research misconduct, and 15% said they'd seen it in others. Again, I think this is very much a minimum estimate, but the numbers are getting larger. I think perhaps we should pause for a moment and think that probably all these things here are just perhaps catching the really inept fraudsters. Cases like this, which are bound to be discovered if anybody looks at the publications. So perhaps very much we're looking at the tip of the iceberg. I don't want to give you a tutorial on how to commit fraud, but if you do want to do it, there are much cleverer ways to do it than what has been done here. For example, I'm not sure how it would ever get found out if you did a real experiment, but just with different antibodies or different drugs. It would be much harder to find that out. Perhaps we can talk about that later. So something that interests me is, why do people commit fraud? Well, one explanation, though it's not really an explanation, is that these are just flawed individuals in the same ways that outside the scientific arena, people commit crimes. Perhaps they're flawed and do that. But I think there are other reasons why even an average person might commit fraud. And one is pressure. Think of the pressures put upon people of their scientific careers. Imagine at the start of your scientific career, you're a new graduate student or postdoc in a lab. Your predecessors have done very well. They've had papers published in the most prestigious journals, and your PI or supervisor was therefore very impressed by them. Your research isn't producing such great data, and your PI therefore is less impressed with you. I think it's pressures like this might make people invent data just to impress their PI. There are more direct pressures, one of which is financial. In several countries in the world, when you publish papers in top journals, and these are journals with high impact factors, individual researchers are given cash. And you can find an article about this in Science. And it came to, this is the sums of money found in various countries in the world. Initially, the publicity was all about China, where sums of up to 160,000 US dollars were given to people who published in the top journals, but all sorts of other countries, including the United Kingdom and the United States, people get cash bonuses for publishing in top journals. And really, which of us could say, if you were given a large enough sum of money, this might not influence exactly what you do and exactly what you publish? So I think money for many people can be a stimulus. There are other things as well, one of which is career progression. So let's say you've done a PhD, you've done a postdoc. What you want to do is to apply perhaps for an independent fellowship or a tenure track position. There's no doubt that to get one of those, you need to get publications, and it really helps to get publications in the top journals. So anything you can do to do this, you're likely to do. Other incentives as well, which I'll talk about in a moment, are the world of impact factors. It's not good enough these days just to publish papers in respectable journals. What you really have to do to get taken seriously as a scientist is to publish papers in the top journals with the highest impact factors. And I think this is worth considering a little bit more. This graph here, it's not from the cardiovascular area, it's from infection and immunity, but I'm sure you get the same if you looked at the cardiovascular area. The y-axis is the impact factor of the journals in which papers are published. The x-axis, they called it retraction index. It's basically proportional to the fraction of papers in that journal that are retracted. I think this is one of the best linear regressions that you'll ever see. And what I want you to notice is that the journals up here, which have the highest impact factors, also have the highest fraction of papers rejected. The journals down here with the lowest impact factors have the lowest fraction of papers rejected, and in between here. So this correlation, it's hard, as with all correlations, to work out causality. And there are basically two explanations about cause and effect. One is quite simply that what I call the high rent journals, the high impact factors, the papers are read much more, and therefore fraud is much more likely to be detected. That's hypothesis one. Hypothesis two is if people are going to cheat, there's no point in cheating to publish in a journal like this. If you are going to cheat, you might as well go for broke and publish in the really high rent journals. Now, when I used to give this talk, I used to end up by saying that I can't distinguish between these two explanations. It could be one or the other, although I suspect this one here is at least an important factor. But one of my colleagues drew my attention to a nice paper done in the crystallography world. And it's this one here, which I think answers the question. This is what they did here, Brown and Ramaswamy, was to look at the quality of protein crystal structures, how good the structural characterization was. And they compared it with the average across all journals. And anything with a mark to the right here means that the characterization is better than the average. Anything with the mark to the left here, the characterization is worse. So these journals here, the characterization is better. And these are journals, the European Journal of Biochemistry, Biochemistry from the US. These are standard journals. The ones down here, the characterization is much worse than the average, and these are the high rent journals, Cell, Science, Nature, etc. So what this is saying is that structures as published in the top journals are worse than those in the better journals. And the authors conclude the most striking result is the association between structure quality and the journal in which the structure was first published. The worst offenders are the apparently high impact general science journals. The rush to publish high impact work in the competitive atmosphere may have led to the proliferation of poor quality structures. So I take this therefore to mean that papers published in these top journals are more likely to suffer from problems as a result of some of these issues. So to try and sum up this part of the talk, I think very much that fraud is perhaps a gamble. In the short term, you're going to get some definite benefits if you commit fraud. You're going to get your papers published in high-rent journals. You may get money directly from this as a result of some of the bonuses we've talked about, but you'll certainly, by publishing in these journals, get a lot of scientific kudos and respect. You'll get your first job and you'll get promoted. And I think these are all very much definites. Possibly, in the longer term, you may get exposed and disgraced. But there are many people who've had to retract multiple papers who still continue their scientific careers. And I wonder, therefore, whether subconsciously or consciously, people are looking very much the way our system operates at the definite and short-term advantages of committing fraud, as opposed to the possible longer-term problems. I know it sounds cynical, but I still think this is something worth thinking about. I've highlighted that I think a significant responsibility comes from journals and journal impact factors. And this is not an original comment. I refer you to this editorial written by Richard Ernst, who was one of the developers of MR imaging, who got the Nobel Prize for Chemistry for this. And this lovely editorial he calls The Follies of Citation Indices and Academic Ranking Lists, a brief commentary to bibliometrics as weapons of mass citation. And what Ernst says, and as an ultimate plea, the personal wish of the author remains to send all bibliometrics and its diligent servants to the darkest omnivoric black hole that is known in the entire universe, in order to liberate academia forever from this pestilence. What he's saying is that these impact factors are there as a quick, easy, and lazy way to assess scientists. And he says there is an alternative. Very simply, start reading papers instead of merely rating them by counting citations. And I think this goes well beyond scientific fraud. And as I say, I commend Ernst to you all. Okay, whatever the fraction of papers that are flawed because of fraud, I don't think it's the main cause of lack of reproducibility. I think perhaps the main causes are firstly, uncontrolled experimental factors, and secondly, poor design and/or statistical analysis. And I'd just like to talk about these briefly in the rest of the talk. What do we mean by uncontrolled factors? Well, these are well known. If you work on cell lines, for example, it's now realized that many of these are contaminated, phenotypically altered, i.e. their properties have changed with passage, or indeed incorrectly labeled. There's a famous case of a confusion between a cell line that was thought to be a breast cancer line that turned out to be an ovarian cancer line, of which billions of dollars have been spent on research using it. If you use animals, we're increasingly aware of the importance of strains, different strains of mice, the environment, what sort of light day cycles animals are kept on, and also the diet. And at least in the past, none of these were typically reported in papers. As far as diet is concerned, one of the nicest examples, I think, comes from this lovely paper from Leslie Leinvan's lab. They were interested in the effects of heart failure on the expression of the beta myosin heavy chain. So the original purpose of the experiment was to compare wild type and cardiomyopathic animals. And there was a modest difference, but it was nothing compared to the effects of feeding the animal on a soy diet compared to a casein diet. The effects of just changing the diet were enormously greater than those of heart failure. And I think differences like this may explain differences of people's papers from different laboratories. Genetically modified mice are a very powerful tool in studying mechanisms, but we're all aware today that this work to be interpretable has to be done on identical backgrounds. Otherwise, you could be looking at the products of a gene other than the one you think you're interested in. Chemical probes, finally, what we used to call agonists and antagonists, work using these can be flawed if they're not specific enough or they're having effects off target. I think a major factor responsible for lack of reproducibility is that of the area of scientific design, analysis, and statistics. I think a major problem may be simply that we've already picked all the low-hanging fruit and done the easy experiments which have large effects. And today we're looking, I think, at smaller effects, and in some ways that's important. If we're interested in heart failure and we want it to be clinically relevant, there's no point in looking at the effects of infarcting 99% of the ventricle. What we should be doing is looking at the effects of much smaller infarcts, which are what people typically get. So that's one factor. Another factor is what we're comparing our intervention with. So in the old days, it was easy. This is a sort of typical experiment. It goes back to Sidney Ringer, but when Mike was visiting me at University College London, this is the sort of experiments we were doing. You get a preparation, here a papillary muscle, you do an intervention, you remove calcium, as everybody knows, the force gets less, you add it back, the force recovers. So this is really simple. The same tissue is a control, an intervention, and a recontrol. You almost don't need statistics if you've got something like this. So we're comparing the same tissue. The problem today comes that so much work, we're not comparing on the same tissue, we have to compare between animals. It might be we're comparing a heart failure animal with a control, or we're comparing a transgenic animal with a wild type animal. And this is, I think, where a major problem comes. Let's imagine we've got one mouse and we compare it with an obese mouse which we've produced by some mechanism. Here I've just done it by enlarging the picture, but that's not really relevant. So let's say we're electrophysiologists and we want to look at ion channels in these mice compared to these. So we isolate cells, and perhaps we isolate 8 cells from this mouse and 8 cells from this mouse. The problem comes as to how people do the statistics. Is this n equals 1 because it's n equals 1 animal, or is it n equals 8 because it's n equals 8 cells? And if you look in the best journals, you'll find many cases where people say this is n equals 8, and the problem with doing that is called pseudo replication, because these are not independent samples. They're all taken from the same animal, and they're more sophisticated ways to do this. And I think this is a major problem in much of certainly cellular physiology. What it means, of course, is you may need more animals. This has cost implications, and it also has implications for the current 3Rs drive to reduce the number of animals. But if you don't do it properly, it means you're in danger of wasting animals by doing the statistically invalid things. Another area which many of you will be aware of is that of false negatives. Let's imagine we have a hypothesis that the decrease of force in a muscle, perhaps skeletal muscle, is due to an intracellular acidification, a decrease of pH. So I've invented this data. Here's the average muscle force for our control, and this is from the fatigued animals, and this decrease here is significant compared to the control. So now what we do in this thought experiment is to measure intracellular pH. And when we do that, we find there is no significant difference in the pH of these animals. And what many people will conclude in published work is therefore, because there's no difference of pH, this decrease of force here is not due to a change of intracellular pH. But of course, what we should be worried about is that the experiment is just underpowered. We don't have the precision to measure the changes of pH, the small changes of pH, which can be responsible for this effect here. So false negatives due to underpowered experiments. Equally worrying are false positives. If you want to read about that, I recommend David Cahoon's work on the false discovery rate, and I'll just leave you with one sentence, and he talks about how p-values are grossly misinterpreted. He says all the approaches above suggest that if you use p equals 0.05 as a criterion for claiming you have discovered an effect, you will make a fool of yourself at least 30% of the time. This alone implies that many published claims are not true. I think it's made worse when one has a rigid statistical threshold. As you'll know, we're all taught as undergraduates that P is less than 0.05 is the threshold at which we should determine statistical significance. Well, that may be what we do in the lab, and we're therefore, but the problem with it is you're happy if your data is 0.049, and you're depressed if it's 0.051, but you don't behave like this, I think, in the rest of life. We can't go to shops anymore, or at least not easily, but if you go to the supermarket and you get a piece of meat like this, you'll see there's a best before date on it, the 10th of October, 2019. What does that mean? Do you really think that it's okay to eat this on the 9th of October, but it's going to kill you on the 11th? I don't think you do. You realize there's a long spectrum of many days or months after this that is probably perfectly edible. You don't have the same threshold that we have with statistics. Other issues, briefly, lack of randomization and blinding. If we're doing a heart failure study, we go to the cage and select a mouse for the heart failure and another one for the control. How do we decide which we choose? Do we rigidly randomize them and therefore protect against the worry that perhaps subconsciously we're taking the weaker looking ones for the heart failure study? Our experimenters blinded to the source of tissues. Ideally, when we patch clamp a cell or measure calcium, we shouldn't know whether it's taken from a heart failure or a control animal. That's fine in principle, but in practice, unless you have a very large lab, it's very likely that the people doing the patching may also be the people doing the surgery. There are practical issues there. And this paper here, which you can read afterwards if you like, looks at published work and finds that just over 15% of people report whether they randomized. A much smaller fraction of people here report whether the studies are blinded. So I've talked perhaps negatively about all the problems. What is the way forward? And this is where I really get worried. Ionardi suggests that what we should do is eventually re-study everything that's been done already. Large studies with minimal bias should be performed on research findings that are considered relatively established to see how often they're confirmed. He thinks that several classics will fail the test. Well, that's fine, but do you really want to be the graduate student whose entire PhD consists of just reproducing somebody else's work? I mean, that's something to think about, I think. The NIH a few years ago announced that it planned to enhance reproducibility. And it may also make validation of work compulsory. And you can see in terms of what I've talked about before, why one might want to validate previous work and why you might want to replicate things. But there's a swing against this. Mina Bissell is a very well-respected cancer biologist, and she points out that the push to replicate findings could get in the way of promising research. And as a patch clamper oneself, you can just think about some of the issues. As many of you, or I guess most of you will know, patch clamp was developed by Nair and Sackman in Germany. And imagine what would have happened if their work had to be replicated before it had been published. There's one practical problem, which is when they did it, they were the only people in the world with a patch clamp. They were the only people who knew how to patch clamp, and scientific discovery and their work, I think, would have been delayed by forcing it to be reproduced. So I think areas like that do need thinking about. Just like to sum up then, I think all the things I've talked about are various ways of missing the truth. We've talked about fraud. I've talked about poor experimental design, execution, analysis. And at the beginning of the talk, I talked about what I think we can't possibly stop, which is honest mistakes. But I think very much this is a continuum, and exactly where deliberate fraud ends and poor experimental design begins is something which we could debate for a long time. I think what they all have in common is they potentially waste other scientists' time. And particularly if you're doing clinical research, they can damage or kill. Where fraud and poor experimental design differ clearly is in intent. But for many things in the outside world, when thinking about crime and things, we focus very much on actions rather than on intent. So I think this is also a difficult issue. And in particular, ignorance of a law is no excuse. So to sum up then, if you're using inadequate statistics and you know that those statistics are inadequate, how different is that from actually committing fraud? So if I can conclude then, much of science, I hope I've persuaded you, is not reproducible. Fraud is one of the many causes. Too much pressure on scientists is likely to cause fraud. Journals and impact factors have a major responsibility in this. Other areas include things like uncontrolled factors and faulty design. And very much we have to sort this out ourselves before our funders put unpalatable pressures on us to carry on doing it. If you're interested more in this, I've written an article about this in the Society's journal, the Journal of Molecular and Cellular Cardiology. And I hope nobody will mind if I end with a gratuitous advert. I'm now the editor-in-chief of the Journal of General Physiology, and I thought I'd just advertise that now. Thank you very much.
David, thank you very much for a really thought-provoking lecture. Could I ask everybody out there, please, if you could post, I'm sure David will be willing to answer some questions. If you could post them on the Q&A tab at the bottom, that would be great. Whilst people are thinking of their questions, David, could I just ask you, since your final slide was about your editorial role, and I know that you've previously served as an editor on JMCC, on Journal of Physiology, you've got a lot of editorial experience. And may I put it to you, my lord, that editors are to blame, okay? That we now live in a world that if you cannot tell 100% watertight story with every T crossed and every I dotted, journals will not accept your paper. Now, in the good old days, if you read some of the old papers, and I'm thinking particularly by Louis Katz that I read, published in 1955. Well, no, I went into the lab, I did these experiments, I didn't quite understand them. I talked to Sonnenblick told me this, and, you could publish papers that you did not understand and that there were loose ends to. You can't do that anymore. And I blame the editors for this. So what are we going to do about the fact that fraud is probably encouraged by people leaving data in their filing cabinet that they don't understand, or by manipulating their data in such a way that it makes sense? We need to be able to publish data that doesn't make sense, because that's the honest way. And that's genuinely what we all generate a lot of data that doesn't make sense. What do you think about that?
I couldn't agree with you more. So one of the many things I didn't talk about was publication bias, which I think you've just alluded to, which is it's much easier to get a positive result published than a negative result published. So that's another reason why many of the public positive results out there are probably erroneous, because one lab gets a positive result, it gets published, 10 labs don't get that result, so they don't publish it. I think journals have a responsibility. I think at the end of the day, the author clearly has the major responsibility. Journals have a responsibility. I highlighted the so-called top journals because to get your paper published in one of those journals, you need a really simple story. They don't want equivocations. But no, I think we have to go back to the time when you could say that in X animals, we got this story, but in another Y animals, it was much more complicated and we don't understand it. Thank you.
So just to say that we've actually got Irv Zucker, who is the AJP heart editor. I know he can't speak, but maybe Irv, if you can hear us, maybe you can use the Q&A box to express your opinion on this as well.
That's great, David. We've got a load of Q&A questions. I just should say before we go on to those, my next paper with the stuff that I can't understand will be going to Journal of General Physiology. David, if you call up your Q&A box at
Yeah,
the bottom of your screen.
Okay,
you
Ben Thomas says, with respect to your slide about short and longer term gambling, do you think we should have some sort of blacklist or equivalent as a punitive measure? The problem with a blacklist is sometimes it's not clear exactly who was responsible for the fraud. I mean, you've got a paper with multiple authors. Who is responsible for it? What is the responsibility of the corresponding author? If your collaboration involves work from many laboratories with different techniques, can you really be responsible for every single piece of data in the work? I mean, having said that, on the other hand, what I don't like is the idea today that when a paper is retracted, often it's the postdoc who is blamed, or the PhD student who's blamed by the senior author. Sometimes it may be those people who've committed the fraud, but other times I'm worried that they're being used as a scapegoat. I hope that helps. Rudolf Fischmeister says, in my long scientific career, I've seen fraud on a few occasions. What strikes me most is that even when published data turned out to be clearly fraudulent, there are always some labs who manage to confirm the data. Does fraud stimulate fraud? That's great. I mean, I'm sure you're right that people have an expectation of what they're going to find. I mean, one example, it's not really fraud, but everybody's taught as undergraduates that the value of calcium in resting cells is 100 nanomolar. And it's amazing to me how many people publish data finds 100 nanomolar. And I do wonder whether this is because that's the value we expect to find. I'm not suggesting that's fraudulent, however. Jessica Caldwell, please can you comment on the problem of publishing negative data? Are strides being made in make it able to do this? I often speak to people who have found the negative data and are not able to publish. You're right, lots of journals wouldn't publish negative data, and also wouldn't publish confirmatory data. Today there are many more respectable open access journals. I think the trend was started off by the PLOS journals, who will publish confirmatory data and will publish negative data as well. So I think things are changing. I think you'd still find it hard to publish in the absolutely elite journals a paper which was purely negative data. Jean-Pierre Benetin, David, what could be the cure of fraud? Hydroxychloroquine. Well, there are probably people from the United States watching this, so should we steer clear of that for now? But thank you, Jean-Pierre. Cesare Terracciano, David, what is the solution for this? The solution for the whole fraud thing, I think a major thing is to put less pressure on people. And I think there's much more pressure on young people today than when I was a PhD student. When I was a PhD student, one was delighted to publish one's work in any respectable journal, and the respectable journal were all the mainstream physiology journals. These days, students want to publish papers in the really top journals. There's much more pressure put on them.
David, do you mind if I just interject here? I mean, I would argue that there is as much pressure on the senior academics as there are on the junior academics. Possibly more.
No, absolutely. Sorry. I was comparing being a young person today with a young person then. Now, absolutely now, yes. No, it's just much more competitive today.
I think trying to secure that next £1 million or £1 million grant is probably more of a pressure than trying to get a PhD, I suspect. Well,
the other thing is, this makes me sound like an old man telling stories. When I first got my lectureship, which is equivalent for those outside the UK of an assistant professorship, in those days there were people publishing good science who didn't have a grant at all because the institution gave you money as well. So that whole pressure was much less. James Clark, what, if anything, do you think institutions can do about policing this? Or is it down to the individual academics and journals? James, that's a very important point. The responsibility is with the institutions, not with the journals. So as a journal editor, if somebody reports suspected fraud, you ask the university or the institution to investigate it. The problem with that, of course, is that the institution has a clear conflict of interest. They're employing somebody very prestigious who's bringing great luster to the institution, and the last thing they want is for all that work to be shown as smoke and mirrors and fraudulent. Now, the best universities, and we've had one example of that in my talk, nevertheless do police it properly, but by no means all institutions do. So that's a major question about whose responsibility it is. Mike Curtis says, the talk crashed when David was talking about p-values. Does David think it is better, and in quotes, to report exact p-values? If so, what is the reader expected to infer when authors provide exact p-values without giving a steer in methods, what they themselves infer from p equals 0.03 versus 0.07, etc.? Cahoon is very vague on this and uses p as less than 0.05 when it suits. I think giving p-values, exact p-values, has to be better than just thresholds. Otherwise, you don't know whether it was just 0.049. I'm not a statistician, but people have said rather than using p-values, you should give confidence limits and things. And there's a whole literature on that. It's a complicated area. Julieta Paloemek, what can you suggest about the N of cells and the N of animals? That's a difficult question. I mean, first of all, you need the statistics to be valid, but also I think let's imagine you have an enormous effect. So statistics would show it with 3 animals. I think most of us would feel unhappy publishing something just with 3 animals. You'd need a larger number than that, but I'm not sure that's a statistical answer as opposed to a psychological one. I don't know. Concerning this, Lucas Sala, concerning the slide on lower quality of protein structures in high impact journals, may this discrepancy we see between journals be related to the fact that high impact journals, at least for cardiology, are looking for highly multidisciplinary, high impact mechanistic stories that require such a broad expertise that may be difficult to control for both PIs, reviewers and editors. That indeed, thank you, Luca, is exactly what the authors of that crystallography paper say. That basically, if you take one of these high impact papers in our field, it's got genetics in it, it's got experimental cardiology, patch clamp, molecular biology. No single referee can be expert on everything. If you send the same work or part of it to a mainstream society journal, if you're a physiologist, let's say to the Journal of Physiology or the Journal of General Physiology, you will get expert reviewers on that particular technique who are much better able to judge it. Dan Johnson, I was wondering what your thoughts were on the Registered Reports initiative, which has just been started by PLOS. Do you think this may change things? I think what Dan is referring to is the analogy with the clinical field, where you can only do it before you publish a trial. You have to, in advance, say what you're going to do, and you can only publish questions that you've defined in advance. So the question is, should we be doing that in basic science? I suspect what many of us do in basic science is you decide you're going to study something, the experiment is worthless from that point of view, but comes up with interesting other observations which lead to a hypothesis. What you're supposed to do, I'm told then, is to just then do another set of animals to investigate this new hypothesis. I think that's very difficult given the sort of parlous grant funding we have today, but I mean, I'm sure it's something that will be forced upon us. Irv Zucker, who has just been mentioned. This is getting better, but I continue to see papers that do not show individual data values, especially in bar graphs. Editors and reviewers need to do a better job. At AJP, Heart and Cardiac, we've added a figure reviewer to scrutinize bar graphs and gels. Irv, that's a free advert for you, but I suppose I had one as well. And editors indeed are getting better, and he makes a valid point there that if you just show means and standard errors, you can cover up a lot of very dubious stuff. It's harder to do that if you show the data points. Dr. Daniel Gonzalez, do you think a small lab can really afford a robust experimental design, double blind, etc.? It's harder for a small lab, but you can get a colleague who's not working in your lab to go to the animal house and select the animals for you. You can get an animal technician, someone else to do it. You can get your histology slides looked at by somebody else. Clive Orchard, in some disciplines, the experimental design is reviewed before the experiments are performed. If it is seen as acceptable and the experiments as important, the results, whatever they are, published. Is this a way forward? I think that's Clive, that's exactly the same as what happens in the clinical area and what I think Dan mentioned about pre-registering reports. It is a way forward. I guess the only issue there is, in some areas at least, people will be worried. They have a brainwave one day, they carry out the experiment the next day, they may not want to let everybody know what their clever idea is for doing an experiment. You know, competition gets in the way of that, but otherwise a good point. Sian Harding, who I have enormous respect for, can you reproduce the sponge cake that Mary Berry makes? After all, the instructions are clear and there are only 3 ingredients. For those of you who don't know, Mary Berry has a TV program. Is it British Bake Off, Davil?
Why do you ask me?
Okay. So I think what Dan is saying.
I clearly look like someone that likes cakes.
So one reason that one can't reproduce data is that I may not be as good at doing experiments as someone else is. This actually resonates very well. I'm not going to name who the person is, but there was somebody who's had to retract several papers recently in the cardiovascular area was once challenged at a meeting by someone else who said they could not reproduce Dr. X's work. This person responded by saying, can you sing like Pavarotti? And, you know, clearly saying that the person who was critiquing them was just a second-rate scientist. So I think, yes, we have to be aware of the fact that some people are much better at doing science than others, but that may not be the only reason that you can't have your work produced. But anyway, nice question, Sian.
And sadly, Mary Berry is no longer in British Bake Off.
Okay, sorry, I'm out of it. Mike Curtis, here's another advert. Have you seen British Journal of Pharmacology's guidance on design analysis? I wrote it. The pushback has been immense and the journal has found it a nightmare to get editors and referees to check compliance on randomisation, blinding, etc. My question is, how do you police all this without antagonising people? I think Mike's making an important point. I think the problem is that journals have to compete to get submissions. And if one journal goes out on a limb in being tougher on statistics, people will send their papers to other journals. And I think the whole field has to move together. And I think this is starting to happen now. It's taking a long time, but I hope Mike is happy that his initiative at BJP has been followed by others.
David, could I just interject at this point? We've got a lot of questions to go yet. It's entirely up to you how many you plough your way through. If you're happy to carry on, I'm sure we've still got a good audience and they're happy to listen. So, I mean, what else are you going to do? You can find a home, answer some questions if you're happy, but feel free to stop at any time.
I'm happy, but you pull the plug on me if you get bored, Mike.
No, I'm not bored yet, so carry on. So,
Gary Loper, what are a journal's recourse if they identify fraud in an article submitted? How often do they contact the institution to point out they've identified fraud in one of their members' papers? I think journals are supposed to contact the institution. So what I always used to do, there's a very useful organisation called, I think it's the Committee on Publication Ethics, COPE, who give guidelines about this. So what I always did was, if a reader or a reviewer says they suspect fraud, the first thing you do is you write to the authors, saying this has been drawn to your attention. do they have an explanation? Because sometimes there will be a completely innocent explanation. The wrong diagram got, if it's at the review stage, the wrong figure may have got submitted, etc., etc. If they haven't got a convincing explanation, I think you're honour bound to get in touch with the institution. I hope that helps. Michael Taggart, how much can be explained in relation to the pressures to perform certain research and publish in certain publishing houses by our inadvertent discouragement of curiosity-driven research, especially for young people? I agree 100% with you, Mike. I mean, particularly about publishing in certain journals. Rashayana Bassani, the pressure to publish high-quality papers in quantity and at high-rent journals is a long-standing plague. Much is said about it, but I see few practical proposals. Do you have any to share with us? I don't know, because if a young person like Davor comes up to me and says, where should he publish? Obviously, Davor, you should publish all your papers in Science and Nature, because that's going to help your career. That's the practical advice to you, but it goes against everything I've said. The system has to change. So, Rosanna, that's not a very good answer. Ildiuterus Mendoza from Chile. How do you think the scientific community can encourage opening doors to small scientists who do not commit fraud, but also less likely to publish in high-impact journals? I think the scientific community is very respectful of people who publish in what you call not high-impact journals. I can think of people in my field who are enormously respected for that. I think the problem comes about by the institutions we work in. One problem, perhaps, in this country, many of our universities have been reorganized. We no longer have separate departments of physiology, pharmacology, and biochemistry. We have large multidisciplinary departments, and that means that one is not competent to assess the quality of the works of one's colleagues when it comes to them getting promoted. In the past, I worked in a physiology department, and I could reasonably well assess the quality of work at neuroscience and the kidney, because the conceptual questions were very similar to those I was asking in the heart. That's not the case today. And so I think it's partly a responsibility of our institutions. Anonymous attendee, is there a correlation between fraud and laboratory science? Shouldn't the PIs be held more responsible for things coming out of their labs than not the postdoc that gets thrown under the bus? I don't know about the correlation between fraud and lab size. Some of the famous cases recently of fraud have been large labs, but it may just be they're publishing more papers. That may explain it. Yes, I think PIs have an enormous responsibility, but rather than the postdoc, but equally, if you are a PI and you have a rogue postdoc, can you be held 100% responsible for that? If you've got somebody who is committing fraud in a very sophisticated way, how would I discover that? I mean, I think it's one of the greatest nightmares most of us have, how you would deal with that. Peter Wilmshurst, who, hi Peter. Peter, I recommend, he's written a lot about fraud in the commercial area, dealing with clinical things as well. What about the role of academic institutions in pressuring people so they commit fraud and covering it up to protect the reputation of their institutions? What about people being effectively trained to commit fraud because they work in departments where fraud is actively encouraged, including by department heads? What about the co-workers who put their names on papers that they know is fraudulent? Peter, I've heard you give your presentation, and it was absolutely, I think we should get you to give a talk in this series. Some of the things you talked about clinical cardiac devices were really terrifying, but I think your question answers itself. It's rhetorical, unfortunately. Peter Ferdinandi, one way to minimize fraud and bad study design, regular lab meetings, the presence of all the scientists and students in one's research group. Such a cooperational way of study design, evaluation of interim results, gives transparency. You're absolutely right, unless you've got a real rogue. And of course, it's more difficult with collaborations between labs, but you're absolutely right.
David,
Natalia Torre.
Sorry, David, could I just interject there? Because I think you raised a really important point about pressure on young postdocs, young PhD students, etc. And, you know, I've progressed from when I was a young PhD student and visiting your lab, and now I run my own lab. And one of the things I feel really is incumbent upon me is to not generate pressure on my junior members of staff. And as I said to them, you know, once jokingly, I know you're not making up this data, because if it did, it would make sense. But seriously, you know, actually putting expectations on our juniors, telling them what the result is that we want, in inverted commas, creating pressure to get a particular Western blot or particular result. And then, you know, we all know of people who go off on ballistically in a lab meeting because the juniors have not got the result they want. And so there is a responsibility for us as senior lab heads to make sure that we don't fall into that trap of putting pressure on our juniors. And I think, in a way, Peter's highlighted that by saying, look, regular lab meetings, but how we behave in lab meetings, for me, is massively important. And Davor, you don't need to chip in at this point.
No, actually, I was going to, because I can vouch for what you said. Very often, Mike didn't have a clue about the data that I presented to him. Because
exactly. Anyway, moving on.
No, a very important point, Mike. Thank you very much. Natalia Torres, as I progress in my career, one of the things that worries me most is I am not the person doing the experimental work, and I fear not being able to detect problems with how experiments are done. Which strategies can be used to deal with this? I don't know. That's a real problem. I mean, I think Mike has just addressed that. Lou Roderick, what do you think of the importance of publishing during your PhD? In many countries, publications are required for PhD, others such as the UK not. This is a time point in a career where pressure is extremely high, but their value system may not be so developed. I mean, I think perhaps you're saying there, Lou, that particularly when you're publishing during your PhD, you may be more sensitive to these stresses and problems. I mean, I think, to paraphrase what Mike has just said, that's particularly the time when you need the support of your supervisor and PI.
But, you know, in a lot of Scandinavian countries, I think you need 2 or 3 papers to get through your PhD. And if the data is not publishable, then what do you do? Oh, I see. Oh, yeah. So you're saying that's an additional. That's what Llewellyn is referring to.
Yeah. Oh, sorry. You're saying that's an additional stress on you. Yeah. No, I see that. Yeah. I mean, I don't know in those countries what happens if, presumably, if you get negative results, that should still be okay.
As long as you publish them.
Yeah. Yeah. That comes back to the previous question. Giulio Agnetti, perhaps an international tribunal economically supported by local governments, composed by scientists and divided by fields of science, could be a solution. Banning from PubMed fraudsters for various amounts of time, depending on the gravity. You could do that. The real problem is you might catch, what would your threshold be at which you would ban people, and what about blameless people? It's difficult. Alicia Mattiazzi, I agree with the live answer to Cesare, less pressure is one solution. Also, the corresponding author has to be responsible for the whole paper. This requires time and dedication, and again, the pressure for publishing. I like your point, Alicia. I mean, I'm just not sure what happens in a multi-lab paper, whether, I suppose these days you have several corresponding authors, so perhaps gone is the time when one person could be responsible for everything. Zhongjing Cheng, says rather than p is less than 0.05, should we consider significance only when p is less than 0.01? I think David Cahoon says we may have to have lower cutoffs as well. So I think that's a step in the right direction. Keith Brain, why don't journals report fraud to the funding body as well as the university? That might exert more leverage. That's a good question. I mean, I guess it is the university who employed the member of staff, so they're considered to be responsible for it. And proper universities do it properly. I don't know why the funding body. That's an interesting question that perhaps somebody should consider more, particularly as all papers today have to give an indication of who the funder is. Anonymous, would it be about the animal cell experiments? Would it be correct considering as n the number of animals, but having the same number of cells? Or since we use normal and obese mice, we should use percentage of cells instead of numbers? No, I think the problem there is it's different to, if you're doing one cell each from 5 animals, that is completely different from doing 5 cells from one animal, because if it's a surgical study, differences in the surgery will have effects that differ between animals, but will not have differences on the cells. If you want an analogy, imagine you want to look at human blood pressure. It's great to measure blood pressure from 5 individuals once each. It will be stupid to measure from one person blood pressure 5 times. I hope that helps. Emmanuel Solomonides, pressure on early career researchers to publish is extreme and one of the reasons of less reproducible science. How can we move away from this? We say journal editors are to blame, but most fellowship review boards require senior author papers in high impact factor journals. So we also do this to ourselves. Absolutely right. Do we need to rethink how to assess young researchers? Are open access journals the way to go? Moreover, how can we change the attitude of journals to be less willing to publish negative results? Wow, lots of good questions. I think, yes, we have to rethink how to assess young researchers. I think we have to quote the paper, the editorial I showed you before, we have to do a subjective decision and read people's science and see how good we think it is. I think people don't like subjective things, but assessing the quality of somebody's science is, after all, a subjective activity. What people prefer is objective factors like citations and other metrics. So I think we have to do it subjectively, which is, after all, is what lots of grant panels do.
David, we have about 12 more questions.
Okay.
Could I just ask participants, if you haven't asked your question now already, please stop, because we can go on forever. David, if you're willing to plough on, let's plough on. But if the participants could stop posting questions now, just in the interests of bedtime, that would be great.
How can we change the attitude of journals to be less willing to publish negative results? I mean, I've said journals are the villains. I mean, that's too simplistic. I mean, journals have to compete. And you have to compete in these days of impact factors by getting citations. And papers with negative results on the whole are less likely to get citations. So journals aren't the only villains. It's the culture of the whole scientific community, journals and everybody else, that has to change. Right. That's a clarification from Alicia. Clarification from Peter. David Patterson, should universities mandate training in scientific integrity for students, postdocs and faculty like we do for animal experiments? Mentoring is so important. Absolutely. I mean, definitely. I mean, it comes back to Mike's point about the need to set a good example to your group. I think universities should do this as well. Ursula, thank you for your kind comments. Ben Thomas draws my attention to Elizabeth Bick, who you should come across her on Twitter. She's got a fantastic eye for diagrams, and she's much better than most people are at looking at 2 published figures and seeing they're the same. And I commend her to all of you. Rudolf Fischmeiter, what do you think of PubPeer? PubPeer, if you haven't come across it, is fascinating. People can comment on papers, and it's quite interesting how often people will draw problems with papers, and a few months or a year or 2 later, that paper will get retracted as a result of the community. Sean Harding, sobering talk, David. Generally, I'm delighted I can reproduce my own results and sponge cakes. Here's an interesting point from Josh Goldharbour. Can you comment on the importance and potential conflicts of interest of whistleblowers, laboratory members, for example? Very good point. So it's the same in science as I think it is everywhere else. We are supposed to protect whistleblowers. If you think that somebody in your lab is committing fraud, you should know what to do. You should go to your PI. There must be somebody in your institution who is responsible for this. There'll be a research governance supremo. You should go to them, and the whistleblowers should be protected. The one thing I would say is, in the world of science, the most damaging accusation you can make to someone is that they are committing scientific fraud. So whilst it's very bad to commit scientific fraud, it's equally bad to make an improper accusation of it. So, you know, be careful. But thanks for that, Josh. Peter, yes, that's a good point. Mike Curtis, he's right, but it's negative. Nothing much will change while the journal impact factor is used to measure the value of a paper. Couldn't agree more. So, anonymous attendee, I was wondering if you could comment on fraud in funded grant proposals. Yes, well, one of the cases I showed at the beginning, I think from Duke, I should check, but I think the problem there was fraudulent data in a scientific application that was known to be fraudulent. So we all have, when you write a grant, if you want it to have any chance, you need preliminary data. And the worry is if people's preliminary data is fraudulent. And yes, that's a big problem as well. I think, Mike, you've been very patient. We've got to the end of this list.
Got to the end, only because I stopped them, because I think actually we've got an attentive audience who've been fascinated by what you have to say, and it's clearly something that we're all motivated to try and do something about, but there are some big problems out there. But I think whilst we have a cohort of people who are willing to grasp the nettle and do something about it, then brilliant. Maybe we can end on a positive note. David, thank you so much for an absolutely brilliant webinar. Before we finish, we've got a couple more announcements. First of all, I said at the start that Lucy Carrier would be talking this coming Monday. Sadly, Lucy's had to postpone that, and I believe Rudo Fischmeister is going to step in to replace Lucy, and we'll hear from Lucy later, I hope. Davor, did you want to
say, just to say, because Rudolf Fischmeister sent me the title. So it's Enhancing Cardiac Phosphodiesterase Activity, a Therapeutic Strategy in Heart Failure. And I will announce that in the next round of advertisements as well.
Okay, so David, thank you for your efforts. Davor, thank you for hosting this as ever. Do you want to finish by putting up any further slides, or are you done?
I am done.
Okay.
I can just put up a JMCC slide once this is over.
I'll sort that out on the video, but that's wonderful. Thank you, David, and thank you everybody for tuning into this webinar, and stay safe and stay well.